Hi there, nerdlings. Today in this video, we're going to be discussing the elements of life, and then I'm going to go into a few functional groups and bare bones basics of our classes of biological molecules. Let's get started. So first of all, you need to know that matter cycles throughout the Earth. It cycles throughout all of the different systems, and we have different cycles that different elements cycle through. So we have carbon cycle, we have our hydrogen cycle, water cycle, phosphorus cycle, sulfur cycle, nitrogen cycle. So all the atoms in living systems are common to this planet. And living systems are mostly made up of 6 common elements, and we call those chunks. So we have carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, and a handful of other trace little elements. So the atoms found in biological molecules kind of build on one another when we get to the different classes of those biological molecules. We have really long chains that we call hydrocarbons, and we find a lot of those in lipids. We have hydrogen, carbon, and oxygen, and those are all found in carbohydrates as well as lipids. Now, in carbohydrates, they have a different ratio than they do in our lipids. In carbohydrates, we have a 1:2:1 ratio. We have 1 carbon to 2 hydrogens to 1 oxygen. So 1:2:1. Whereas in our lipids, we have really long chains of these hydrocarbons. So in amino acids, we tack on nitrogen, and amino acids are the building blocks of proteins. Now, in amino acids, we also have sulfur that kind of pops up, and that pops up when we get into our tertiary structure of the protein. Now, when we get into our nucleic acids, DNA and RNA, we add in that phosphorus. And remember that DNA has that sugar phosphate backbone that kind of builds that the nitrogenous bases come out of so they can bond with each other. All right, so carbon is kind of like the big bad mamma jamma of all of our elements. So it is a major structural atom in all of organic molecules. And we call the study of organic molecules carbon-based life forms. So the major abiotic source of carbon is carbon dioxide. And remember, when I use that word abiotic, A means without, and bio means life, so meaning non-living source. So again, atmospheric carbon dioxide, or CO2. We have major biotic processes that involve carbon, and that's when carbon dioxide is actually fixed or incorporated into producers by the process of photosynthesis, and then it's actually returned back into the atmosphere through cellular respiration or decomposition. So if you think about photosynthesis, we are taking the products of cellular respiration, which are water and carbon dioxide. So if I take a deep breath in and I breathe out, your hand is actually a little bit of moist. So that's water, and then the carbon dioxide coming out. So photosynthesis takes that carbon dioxide that I just exhaled, and it fixes it into this wonderful molecule called glucose. Glucose is then used in the process of cellular respiration. It gets broken apart, and most importantly, it forms ATP, which is our energy currency throughout all of our cells. So why do we study carbon? Well, we kind of want to know why we're living and what makes us tick. And carbon is basically the study of organic molecules or carbon-based life forms. So all life is built on carbon. Within cells in our body, they're composed of around 72 to 75% water. 25% of our cells are actually composed of carbon-based organic molecules, whether it be lipids, carbohydrates, nucleic acids, or proteins. Those are all composed of carbon with other elements involved. We also have a few salts sprinkled in there. So carbon atoms are extremely versatile building blocks. They make 4 stable covalent bonds. And remember, carrying is sharing, and sharing is covalent. So that means that they're sharing electrons. And they can make single, double, or even triple bonds. So next up, is hydrogen. This is, again, another major atom in all organic molecules. However, it doesn't exist freely in nature, so you don't typically see just hydrogen kind of floating around. We have major abiotic sources, again, abiotic meaning non-living, and that comes in the form of water, which is found in the hydrosphere. Now, in biotic or living processes and cycles, it's incorporated into all of our food chains and food webs by the uptake of water, drinking, and it's released through decomposition. So we have these really long chains that we call hydrocarbons, and as you probably guessed, it's when hydrogens and carbons are bonded together and we form these long chains. Now, these long chains are non-polar, meaning that they are hydrophobic or water-fearing / water-hating. They will not dissolve in water. So again, if you've ever poured oil in a beaker of water, they don't combine. So these hydrocarbons help to form up some of our lipids or those hydrocarbon tails. And again, they're not soluble in water because they are hydrophobic. This also classifies them as non-polar. They're very stable, so it allows them to form these really long strands, and they grow into very large macromolecules. Next up is nitrogen. So this is, again, a major component in biological molecules, especially proteins and nucleic acids. So in the abiotic source, we find it in the atmosphere as N2. And then for our major biotic processes, we have the nitrogen cycle. We have fixed nitrogen that gets incorporated into our food chains by nitrogen fixing bacteria, and it's then released into the world or to the ecosphere through the process of decomposition as well as denitrification. So nitrogen has a couple of little functional groups. We have what we call as an amino group. And if you guessed it, amino groups are found in amino acids, which are the building blocks of proteins. And they're composed of a nitrogen and 2 hydrogens. We also find nitrogen in our nitrogenous bases or our nucleotide bases that compose DNA and RNA. So we find them in adenine, guanine, cytosine, thymine, and if you're an RNA fan, in uracil. So next up is oxygen, a major component of all organic molecules. And again, the abiotic source is going to be oxygen that we find in our atmosphere all around us. And the biotic process that it is involved with is used in cellular respiration, and it's released by the process of photosynthesis. So again, in photosynthesis, it's taking the products of cellular respiration, which are carbon dioxide, water, and actually ATP in the form of energy, because photosynthesis requires energy to build glucose, which is a macromolecule. So in photosynthesis, we're taking that carbon dioxide and that water, we're fixing the carbon dioxide into a glucose molecule, and oxygen is just a byproduct. So in oxygen, we have several different functional groups that can form. We have a hydroxyl group, which is denoted by OH, and this is classified as alcohols. We have a carbonyl group, which is classified by carbon double bonded to an oxygen. Now, someday you may be in an organic chemistry class and you're going to learn all about aldehydes and ketones and a bunch of other crazy things. But just for little giggles, an aldehyde, if you see that carbonyl group and it's at the end of a molecule, you're pretty much looking at an aldehyde. If it's in the middle, you'd classify it as a ketone. We also have carboxyl groups, which is a COOH or a C double bonded to an O, and then to an OH group or a hydroxyl group. And you find these a lot in fatty acids and amino acids. So next up, phosphorus and sulfur. So both of these have major roles in biological molecules. Sulfur is found in proteins, and then phosphorus is predominantly found in our nucleic acids. Phosphate groups are used in energy molecules such as ATP. We utilize that energy by cleaving off the last phosphate and that high energy bond, and it releases energy that our cells can use. So the major abiotic source for these is the lithosphere in rocks, and it's released by that natural weathering process. In biotic processes that involve our phosphorus and our sulfur, it's incorporated into the food chain from the soil, and then it's released by decomposition. So phosphorus has a functional group that's called a phosphate. And hopefully this sounds familiar to you because we have a phosphate sugar backbone in our DNA molecules. Phosphate is very highly reactive and it transfers energy. And again, think about those ATP molecules. We have adenosine triphosphate, meaning 3 phosphate groups. And when that last one is cleaved off, it releases a large amount of energy and it fuels the cell. It's also found in nucleic acids and certain lipids. We also have a sulfhydryl group, which is denoted by SH, or sulfur and hydrogen, and it stabilizes the tertiary structure of proteins. So, if you remember back to some biochem from maybe your freshman year, I used these guys, little tangle beads, and when we talked about the structures, we had primary structure, we had secondary structure of proteins, which is our alpha helices or our beta pleated sheets, and those are more so held together by our weak hydrogen bonds. Then we get into those tertiary structures. That's where we get those disulfide bridges. So we have some strong ionic bonds, and then we have stronger covalent bonds in the form of those disulfide bridges. So next up, now that we learned about all of the important elements that kind of go into these biological macromolecules, now we're going to briefly discuss those biological molecules before we go in depth with each of those classifications in different videos. So we have 4 different classes. We have carbohydrates. Those are our sugars, pastas. They form cellulose, chitin, starches. They're our short-term energy supply. So if you're really lethargic, if you drink something sugary, or you play sports and you're drinking Gatorade, or you eat that gooey stuff, if you're running a marathon, it gives you that short energy burst so you can kind of get through that. They also serve for some structural supports and the plasma membrane. We also have our lipids, which are our fats, our oils, and our waxes, as well as steroid and hormones. These are long-term energy stores. So if I was stranded on a desert island, the more fat that I have on my body, the better, because then I could dip into that excess storage and use it as fuel. Now, lipids are very important for our cell membrane. So if you think back to cell biology a little bit, we have a phospholipid bilayer. So obviously we need those fats to compose that plasma membrane. It also serves for structure, cell signaling, as well as insulation. And then we have proteins. Now those function in many cellular processes, and they're the building blocks of cells. So proteins basically are needed for every single reaction that goes in our body, because proteins encompass that class of enzymes. And enzymes are responsible for all of those metabolic processes. So whether we are breaking components down or whether we're building things together for storage, we're using those proteins. Then we have nucleic acid, and that's what composes DNA and RNA, and that's how we transmit genetic material. So how are these macromolecules made? Well, we have different little pieces. So we have monomers that come together through processes, and those create polymers, or sometimes dimers if we only have 2. So a monomer is an individual subunit, or a building block, if you will. A dimer is 2, and then a polymer, poly means many. So many monomers placed together forms a polymer. So there are 2 processes that I want to discuss with you before we kind of move on in our lecture. And again, we're going to be using these handy-dandy tangle beads, as well as some chopped up pool noodles. So we have 2 different reactions that we use, or 2 different processes that we use to either build or break biological molecules. If we want to break a biological molecule, we're going to use a process that's called hydrolysis. Hydro means water, and lysis, hacha, means to split. So if these were each a monomer, so if each one of these little tangle beads was a monomer, and I wanted to split it apart, I would use the process of hydrolysis by adding water. So I add water, and that is the process of hydrolysis, splitting with water. That's how we break molecules apart. This is what happens when we digest our food. So I might eat a donut or a cheeseburger, and my body has to break that down so it can utilize what little nutrients there are in a cheeseburger and a donut, which lots of glucose, so I can get some energy, but it has to use that process of hydrolysis. Now, if I needed to put something together, like I needed to store some energy for later, maybe in my love handles, then I need to use a separate process. The second process that we're going to talk about briefly is called dehydration synthesis. So dehydration is removing water, and synthesis means put together. So if I want to bond monomers together or smaller polymers to form a giant polymer, I'm going to use the process of dehydration synthesis. So I'm going to remove water and bond that together. Now, next up, let's make it a little bit more complicated. So say this is an enzyme. This is my substrate, all right? So enzyme binds with substrate, substrate enzyme complex. Now, if this is a large macromolecule and I want to break it apart because I need to digest it and I need to be able to use those nutrients within my body, I'm going to bind with an enzyme. This enzyme is then going to use the process of hydrolysis, hydro meaning water, and lysis meaning hydra, to split. So it's going to use the process of hydrolysis, bam, and now it's split apart. Now this is called a catabolic enzymatic reaction, meaning this enzyme used the process of hydrolysis to split, hydra, the water apart. And the way I always like to remember this is that cats scratch things apart. So a catabolic reaction breaks apart monomers using the process of hydrolysis. So hydro, meaning water, lysis, hydra, to split, and then catabolic, cats, rare, they scratch things apart. So hydra and rare go together. Those are breaking those polymers apart. Now the opposite of that process is called dehydration synthesis. And if we want to put these together, so say I need to store some fat and I need to put some monomers together. So enzyme comes along, binds with these two substrates, substrate complex forms, enzyme substrate complex forms, hocus pocus happens. And now this enzyme used the process of dehydration synthesis. Dehydrate means to remove the water, and synthesis, put them together. Now this is called an anabolic enzymatic reaction. So remember, Anna adds, added these two together, and cats, rare, they scratch things apart. So I hope those visuals using my very exciting cool noodles and my tangle bead helped you understand those two processes a little bit. So again, we have two types. We have anabolic and Anna adds. So taking those two little monomers together and making it a polymer or a larger macromolecule for storage. That's anabolic, Anna adds. And it does that by using the process of dehydration synthesis, removing water and synthesis, putting things together. And then the opposite of that is a catabolic, rare. Remember, cats scratch things apart, and that uses the process of hydrolysis, hydro, meaning water, and lysis, hacha, meaning to split. So whenever I use an anabolic process, I'm adding things together, such as creating glucose in the process of photosynthesis. That is an anabolic reaction, and it consumes energy. Now, the opposite of that is cellular respiration. Cellular respiration is a catabolic reaction. Remember, cats, rare, they scratch things apart. So it's breaking those bonds using the process of hydrolysis, or hydro meaning water, and lysis, hacha, to split it apart. And that is actually releasing energy because we're breaking those bonds of glucose to form our ATP. So just to kind of recap, dehydration synthesis is the process by which monomers of biological molecules are bonded together. and this process does require enzymes. One monomer will lose a hydroxyl group, and the other monomer will lose a hydrogen atom. And these will combine to form H2O, or water. The removal of water forms a covalent bond between those two monomers. And again, this is classified as an anabolic enzymatic reaction because ana adds. And again, the opposite of that is called hydrolysis. Hydro means water, and lysis means hacha, to split. Hydrolysis is the process by which those polymers are broken down into monomers, and it does require enzymes and water. The enzyme helps to break the bonds in the water and the covalent bond between the monomers. One monomer will receive that hydroxyl group, and the other monomer will be left with that hydrogen atom. And this is a rare catabolic process, meaning those bonds are broken using that process of hydrolysis. So I hope that was helpful. Stay tuned for more in-depth videos on our biological molecules, and see you guys next time.